Welcome back to the Learning Studio. This is the Biological Sciences series, and today we're exploring one of the most revolutionary tools ever developed in biology, genome editing with CRISPR-Cas9 and Beyond. If you've missed our earlier genetics and genomics episodes, like Mendelian inheritance and population genetics, you can find the full playlist linked below in the description. Here's a thought. What if we could cure genetic diseases by simply correcting a single spelling mistake in DNA? Or design crops that can survive climate change? Or even bring back extinct species? Thanks to genome editing, these possibilities are no longer science fiction. They're becoming scientific reality. What is genome editing? Genome editing refers to technologies that allow scientists to change the DNA sequence inside living organisms. Instead of waiting for mutations to happen randomly, we can now add, remove, or modify genes at precise locations in the genome. The most famous and powerful tool for this is CRISPR-Cas9. How CRISPR-Cas9 works. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Originally discovered in bacteria as a defense system against viruses, it has been adapted as a gene editing tool. Here's how it works in simple steps. A guide RNA is designed to match a specific DNA sequence. The Cas9 enzyme acts like molecular scissors, cutting the DNA at that exact spot. The cell's natural repair mechanisms kick in, allowing scientists to disable a gene, insert new DNA, or correct an error. It's precise, efficient, and far cheaper than earlier gene editing technologies. While Cas9 is the star, other innovations are emerging. Base editing allows single-letter changes in DNA without cutting both strands. Prime editing, a search and replace tool for DNA. CRISPR-Cas12 and Cas13, targeting DNA or RNA for new medical applications. The field is evolving rapidly, opening possibilities we could only dream of a decade ago. Case studies and applications. Case study 1, sickle cell disease. CRISPR has been used in clinical trials to correct the faulty hemoglobin gene. Case study 2, agriculture. Scientists are engineering drought-resistant crops to address food security. Case study 3, conservation biology. Gene editing is being explored to protect endangered species or even revive extinct ones like the woolly mammoth. Why it matters in genomics. Genome editing represents the shift from studying genes to engineering them. It holds promise for curing inherited diseases, improving global health, revolutionizing agriculture, and even addressing climate challenges. But it also raises ethical questions. How far should we go? Should we edit embryos? Could this technology create inequalities between people? These are challenges science and society must face together. Past, present, and future relevance. In the past, scientists could only observe inheritance. In the present, CRISPR gives us the power to edit life itself. In the future, genome editing may shape medicine, agriculture, conservation, and perhaps even human evolution. Why watch this playlist? This playlist covers the 30 most important topics in biological sciences, with real-world examples and clear explanations. Whether you're a student, teacher, or curious learner, this series gives you the foundation to understand both classical biology and cutting-edge science. So that's our introduction to genome editing with CRISPR-Cas9 and Beyond, the tool that's transforming biology from a science of discovery into a science of design. In our next lecture, we'll look at one of the greatest achievements in modern biology, the Human Genome Project and genomic medicine, how mapping our DNA unlocked a new era of personalized healthcare. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the next episode in the genetics and genomics section. Because once you understand genome editing, you begin to see the future of life itself.